Yes.
Okay, great.
Oh, what do I have
Welcome.
on my
Good morning.
screen now?
Just say
Got
okay.
it. Got it. Oh, okay.
And then Ben, you can go ahead and mute everyone with the exception of Louis after I get done. Good morning, good afternoon, everyone. It's my pleasure to kick off another Sorghum and Millet Innovation Lab virtual seminar. Today we have the pleasure of listening to Louis Prom, who will take lead for the team in Niger and Senegal to describe the results of some of their survey work on the prevalence and incidence and severity of leaf and foliar diseases in West Africa. And project is broadly defined to provide input into on those topics for the breeding programs to develop that, hopefully develop that host plant resistance against the most prevalent diseases that are affecting production in Senegal and Niger. So with that, I'm going to turn this over to Dr. Prom, and he will lead us in the presentations. How about we hold the questions until the end of the seminar, and when you have a question, even if it's along the way, you can go ahead and feed it into the chat box. I'll take a note of that, facilitate the discussion at the end. So Dr. Prom.
Thank you, Dr. Dalton.
Thank you.
This SMEL project, enabling marker-assisted selection for sorghum disease resistance in Niger and Senegal, is led by Dr. Magill, Department of Plant Pathology and Microbiology, Texas A&M University, College Station, Texas. In Senegal, Mame Penda Sarr Diawara, who is a plant pathologist, is the co-PI, and Cyril Jata, a sorghum breeder, is a collaborator. In Niger, we have Dr. Adamou in Niamey, who is the co-PI, and Adamou Issa from the University of Deliberi, who is a collaborator. This project aims to identify resistant sources to anthracnose, grain mole, and long smut, whereas in Senegal, our concentration is on anthracnose and grain mole. The first step to identifying or effectively managing disease is to have a knowledge of the occurrence, the distribution, and the relative importance of the particular disease you want to work on. In this project, one of our sub-objectives is to conduct multiple disease survey in production fields in both countries. Today, I'll be talking about the survey that we conducted in Niger, then Senegal, then why are these surveys important? And finally, discuss what I consider to be essential future priority research. The survey in Niger. In Niger, 121 fields were surveyed from 4 of the 5 major sorghum producing regions of Deliberi, Tawa, Dosso, and Maradi. This survey was conducted on major production fields that were about 30 kilometers apart. At each of these stops, we surveyed 2 to 3 sorghum fields. These sorghum fields were planted mainly with the land races. However, there were a few that were planted with improved cultivars released by the National Institute for Agriculture. In each field, 60 plants were assessed using the W-shaped pattern. The W-shaped pattern of surveying diseases on production field is the most effective way to actually cover the entire field. In each field, what we did was record the incidence of each disease of sorghum. At the end of the survey, we calculated the prevalent rate and also the incidence of the diseases that we encountered during the survey the prevalent rate of the disease was calculated based on the number of fields with that particular disease over the total number of fields that we surveyed multiplied by 100. For the incidence, is the number of plants with that particular disease over the total number of plants that was accessed in that field multiplied by 100. So that gave us the prevalent, how widespread is the disease, and also the incident, how bad is the particular disease in the individual fields. Oh, it's not going down. Can Benjamin help? The slide is not moving. Yeah, the screen is not moving. Can Benjamin move the next slide? Can you move the next slide?
I think you might have to
get the IT guy.
Well, it's not controllable from our side.
Okay,
but,
it's supposed
if
to be
you
controllable from here.
If you go to your PowerPoint and do you know where your PowerPoint program is?
No, it's not here. It's the wide screen that I'm using currently right now.
If you
But I
escape,
can call the IT. It's just
Go ahead
just call the IT guy to come in. You can just pause it.
I think if you hit the escape button, you should be able to get back.
Yeah, it's not moving. Let me try the escape button and see. Gabe, it's not moving.
Yeah, all right, thanks.
And you have a copy of the presentation, so why don't you just stop sharing your screen, Louis, and then you can load it up.
I've got a copy too.
Yeah,
And you
he'll
can
be here in a
control it
minute.
then.
You can just
wanna
pause
try-
it.
You want to try that?
I don't know why it's not moving.
Yeah, let's see.
I was using this one.
Okay, so close it down. Just escape.
Escape.
You close it.
Right. Well, continue. Will it go? Okay, this slide shows the map of the regions that we are surveyed in Niger. We have the region of Tawa, Teleberry in the northern part. Then on the eastern part of the slide, on your eastern part, we have Maradi, and on the south, we have Doso. Oh, it's not, Gabe, it's still not moving. Oh, Dr. Magill, okay.
Yeah.
Thanks. Dr. Magill can go ahead and move the slides for me.
Yeah,
Okay,
yeah.
the results. Okay, these are the diseases that we encountered in both countries, in Niger and Senegal. If you look at the first picture, that's the leaf blight, showing you the elongated structure surrounded by either a purple, tan, or red coloring based on the cultivar or the plant color that you are dealing with. And the second picture is an infected leaf of anthracnose. You can clearly see those black dots, which are the fruiting bodies or the esaboli on the infected leaf, anthracnose infected leaf. The next picture is an infected midrib of sorghum. And the final picture on your right-hand side is zonate leaf spot with the concentric ring that you can see there. For the first, the infected leaf of rough leaf spot is on the bottom row on your left-hand side. This can easily be distinguished from anthracnose infected leaf because the rough leaf symptoms have a sandpapery feel. So if you touch it with your 2 fingers, it will be sandpapery. So that differentiates it also from anthracnose, where you have the esaboli. In the rough leaf spot, instead of esaboli, you have pycnidia, the raised pycnidia that give it that papery, sandpapery feel when you touch the leaf. The next picture on the bottom, second row, is a long smut. The long smut of sorghum is slightly different from the other type of smuts. The long smut gives you a long elongated sac, curved sac, that is really distinguishable from its closest relative, like the cover smut, which is the next slide. You can check the cover smut. That doesn't give you that elongated symptoms. The final symptoms, the final disease that I'm looking at here is the overleaf spot. The overleaf spot, you have a spot on the leaf, And surrounding that spot, you have either a purple, here again, depending on the plant color, you may have a purple or a red coloration. Next slide. Okay, the results. In Niger,
Which one did I go too far?
yes,
Yeah.
yes. No, that's, you've gone too far. Okay, let's, this is the, this actually gives us the average sorghum yield for the different areas, different regions in Niger that we surveyed. You have Telaberi, Tawa, Doso, and Maradi. If you look at the average yield for sorghum in these regions, it's very low. The highest is Doso, which receives also the most rain in Niger. We also gave the type of production systems that they use in the different regions in Niger. You have monoculture in Telaberi, where you have millet, sorghum, then cowpea. Whereas in Tawa, they have intercropping, they intercrop early and late maturing varieties, as well as rotating millet, sorghum, and cowpea. In Doso, similar, they included peanut in their surveys. And finally, in Maradi, they included hibiscus in their rotation. In Niger, 21 different sorghum diseases were encountered during the survey in 2019. Anthracnose was the most prevalent disease in Niger. So the 99% prevalent of anthracnose translate to 120, which means in Niger, where we surveyed 121 fields, 120 of those fields were infected with anthracnose. That's a tremendous amount. What this tells us is that the land races and the improved varieties that they've released in Niger are also susceptible to anthracnose. The second most prevalent disease in Niger during the survey was leaf blight. And 108 of those fields out of the 121 fields surveyed in Niger were susceptible to hard leaf blight. After leaf blight, we have overleaf spot. Overleaf spot was the next most prevalent disease during the survey, which accounted 76% prevalent, account for about 92 fields out of the 121 fields that were surveyed. Let's look at our responsibility for Niger. The Niger, what we are looking for in Niger is resistant sources to anthracnose, grain mold, and long smut. So long smut, we had about 76 fields out of the 121 fields that were surveyed had long smut. Long smut was observed in all the regions, the 4 regions that were surveyed. Now, the other thing that I wanted to look at is the grain mold. We look at grain mold, the prevalent of grain mold was 21%. This translates to 25 fields out of the 121 fields that were surveyed. This number is extremely low. The reason why the number is low is because of our survey timing. The plants that we surveyed were at the late flowering to early hard dose stage. This is not an ideal stage to survey for grain mold. The ideal stage to survey for grain mold is either at maturity or during harvest time. So the number does not reflect really the prevalence of grain mold in Niger. Next slide. Here we saw the incidence of the different diseases that we observe in Niger. The incident for grain mold, the incident for anthracnose was highest in Maradi and Doso. These are the 2 regions that received the most rain. Tawa and Telaberi are much drier. So the incidence of anthracnose was actually higher in places where you have higher rainfall during the growing season. In the case of long smut, the other disease that we are responsible for, you can see that the incidence was higher in the 3 out of the 4 regions that were surveyed. For grain mold, similarly, we've seen that in 3 of the 4 regions that were surveyed, grain mold was at least not extremely high, but at least considerably higher than what we saw in Teleberry. Okay, this slide shows the incidence of leaf blight. Leaf blight was more prevalent in Maradi than the other 3 regions that we surveyed. And in Maradi, 2 fields, the 113 and 115, had 100% incidence. This shows us that if you want to screen for resistance, these are an ideal hotspot where you can get resistance, where you can screen for resistance to leaf blight. At least during the survey in Niger, at least one field in each of the 4 major regions that we surveyed had incidence of 100% anthracnose infection. We have field number 34 in Teleberry, and we have field number 66 and 71 in Doso. In Tawa, we have field 76 and 86, whereas in Maradi, we have field 101. These are ideal spots where anthracnose resistant evaluation can be conducted. In Senegal, our survey was conducted in 206 fields. These 206 fields, the protocol that we use in Niger was the same protocol for surveying that we use in Senegal, except for the fact that in Senegal, instead of assessing 60 plants per field, we just assess 40 plants per field. Most of the fields in Senegal were also planted with land races. However, a fair number of the fields were planted with improved varieties released by ISRA and CNRA. The similar pattern of survey was conducted in Senegal. We used the W-shaped pattern. We also stopped at 30 kilometers and surveyed 2 or 3 fields within that stop. This shows us the map of Senegal, actually. Northern Senegal is much drier than southern Senegal. So the regions that we surveyed were 7 regions in Senegal. The regions with the least rainfall was Thiès, which is located in northern Senegal. And the rainfall annually during the growing season is around 380 millimeters, compared to Kolda, which is the wettest part of Senegal, which experiences rains of over almost 1,000 millimeters annually. This, we also identify some of the locations by the star, some of the locations that we surveyed during the 2019 trip. The results in Senegal. Unlike Niger, where we observed 21 different diseases of sorghum, in Senegal, only 15 diseases were identified across the 206 fields, just 15. And in Senegal, the most prevalent disease in Senegal was leaf blight. And out of the 206 fields that we surveyed, leaf blight was observed in 198 fields. The second most prevalent disease in Senegal was anthracnose. Anthracnose was observed in 150 fields out of the 206 fields that were surveyed. This was followed by zonate leaf spot, which we encountered in 126 fields across the surveyed regions. Here again, we look at grain mole. Grain mole 2% prevalent. It doesn't reflect. This number is actually misleading because 2% prevalent of grain mole in Senegal doesn't make any sense. That tells you that the survey period is very critical. Otherwise, you'll miss some sorghum diseases. If the survey were done at maturity or close to harvest, this number would have been totally different. But based on the timing of our survey, we definitely did not capture the importance of grain mold disease on sorghum in Senegal. Long spot, okay, long spot. Let's look at long spot, another disease that I thought was one of the Panicle diseases that I thought that may be prevalent in Senegal. We just did not see that much. Out of the 7 regions that we surveyed, long spot was concentrated. Where we observed long spot was on the northern part of the country, which is much drier and at a lower distance. Out of the 206, long spot was observed only in about 21 fields. So it's very low. For leaf blight, however, in the region of Kolda, Kaula, and Fatigue, all the fields that were surveyed in these 3 regions were infected with leaf blight. In Kolda, we surveyed 51 fields. All the 51 fields had leaf blight. As for grain mold, the next most prevalent disease that we found in Senegal, it was really also widespread. But in Senegal, out of the 206 fields, we found anthracnose in 140 fields. So that's also significant when you look at it. And in all the regions, you know, apart from fatigue, the reason why we have a 0 anthracnose in fatigue, if you look at fatigue in Senegal, the fields that we just surveyed 6 fields in fatigue, and those fields were planted with improved varieties. None of the fields that we surveyed in fatigue was land raised. So you, at the end of the, later in the discussion or later in the, I'll talk about why we are seeing less anthracnose in some of those lines. So the next objective for Senegal was grain mold. When you look at grain mold at the bottom line, the prevalence of grain mold in these regions is extremely, extremely low. Here again is the survey period, the time that we surveyed the plants. They were just too young. So the infection was not it was not visible. Later in the season, I think we would have had higher levels of grain mold, especially in the advanced, in the improved varieties, because the few fields that we surveyed that had improved varieties, we had a lot of grain mold. The next disease, apart from leaf blight, anthracnose, the next most prevalent disease in Senegal across the surveyed region was zone-8 leaf spot. And zone-8 leaf spot was most prevalent in Tambacounda. Tambacounda, we surveyed 39 fields in Tambacounda. Out of the 39 fields, 38 fields were infected with zone-8 leaf spot. Next slide. These are interesting locations and regions during the survey that we encountered. For instance, in Kolda, we surveyed 51 fields for sorghum diseases. Out of the 51 fields that we surveyed in Kolda, 20 of those fields had 100% leaf blight incidence, irrespective of whether they were planted with advanced cultivars or they were planted with land races. Every single 20 of those fields were actually infected with leaf blight. Similarly, when we look at anthracnose in the next slide, out of these 51 fields that we surveyed in Kolda, 7 of those fields had 100% incidence of anthracnose, irrespective of whether they were planted with a land race or the advanced cultivar or advanced lines. Next slide. Sooty stripe is another disease. The reason why I included sooty stripe is not one of our responsibility to look for resistant sources, but later on it will become evident why I included sooty stripe here. The most prevalent region, sooty stripe was most prevalent in the region of fatigue. However, the slide here shows that 2 fields in the region of Chess and Kaffrine had the highest incidence of sooty stripe. For zonate leaf spot, the most prevalent disease that we found in Tambacounda was zonate leaf spot. As I said, we surveyed 39 fields in Tambacounda, but 38 of them were infected with zonate leaf spot. And these 4 fields, all located in Tambacounda, saw the highest incidence in each of these fields. In field number 78, we had 98% incidence of zonate leaf spot. And in field 105, we had 90% zonate leaf spot in that field. Significant. Why is it significant to do this survey? As I said, in order to determine the priority working with certain diseases, you have to have a knowledge of what's out there. What are the occurrence? What are the distribution of these sorghum diseases? And what are their relative economic importance in that particular country? So when we did this survey, this is what we came up with. In Niger, anthracnose, leaf blight, overleaf spot, roughleaf spot were the most important foliar diseases. As I said, in Niger, anthracnose was found out of the 121 fields that we surveyed. Anthracnose was found in 120 fields. That's a tremendous high district, high number, and will tell you the relative importance of that particular disease. As for leaf blight, out of the 121 that we surveyed, 108 were infected with leaf blight. So that disease is also important. For overleaf spot, 92 fields out of the 121 were infected with overleaf spot. So if you have resistant source for anthracnose, that's what this project requires. One of the aim of this project to get a resistant source to anthracnose. So what you gain by having a resistant in anthracnose, you actually lose by having that same line being susceptible to leaf blight, overleaf spot, or roughleaf spot. So this survey was really significant that way, that you can look at it and say, okay, the project asks for resistant sources to anthracnose. If I have a resistant source to anthracnose, but that source is susceptible to leaf blight, overleaf spot, and roughleaf spot, it just doesn't give us any gain. The other thing that we did before heading to Niger, the set of differentials for anthracnose that can delimit the races or the pathotype of anthracnose, those seeds were sent to Niger so that they can be planted in 2 locations. One set was planted in Telaberi, which is much drier, and another set was planted in Maradi. And what we found from our survey when we assessed the disease in those 2 nurseries, we found out that the pathotype of anthracnose that exists in Maradi is totally different from the one that exists in Telaberi. So that was significant. The other disease that we had to work on to look for resistant source is long smut. In Niger, across regions, long smut was found everywhere. So out of the 121 fields that we surveyed, 76 fields were infected with long smut. In Senegal, here again, I looked at it in a different light. In Senegal, the good news in Senegal is that most of the improved varieties that were released by their national programs were resistant to anthracnose. So I don't know, which means over the years, they've actually looked at anthracnose and say, well, anthracnose is very important. So let's have, in our improved varieties, resistant sources to anthracnose. So the majority of the improved varieties released by their national program is resistant. Those cultivars are resistant to anthracnose. However, those cultivars were highly susceptible to leaf blight and to sooty stripe. That's why I included that graph of sooty stripe. So what happened here? That sense of collaboration we are releasing sources of resistance, but you neglected that there are other sorghum diseases. So this survey is essential to show that, yes, we have resistant anthracnose lines in our improved varieties, but the reverse side of that is that those varieties are susceptible to leaf blight and to zonate and to sooty stripe. So you don't have any gain. You just don't have any gain. So the resistance has to be a combination of different sorghum diseases based on the survey that we did, be able to document what is out there. The good news is that we were able to identify hotspots in both countries where you actually can do resistance screening for these different diseases. You can go actually in Senegal, we have the coordinates, we have everything, as well as Niger, where you can go and say, okay, these are our sources. We actually need resistant sources. Let's go ahead and evaluate them in this part of Niger. The other thing is that this is the documents that we generated from the survey are the first documents of extensive survey of sorghum diseases in these two countries, in both Senegal and Niger. So this documentation will be very important in future work and in future trying to look for resistant sources. You can go and refer to these documents as a guide to where those priorities should be. As I will discuss in the next slide about looking at priorities, future priorities. Do you have that slide, Dr. Magill? Next slide. Do you have the next slide?
No, it doesn't go to the
Oh,
next.
okay, yeah. It's the amended this thing. I think Benjamin has it.
Or you may just have to talk about it.
Yes.
Yeah,
The next slide.
I'll see if I can pull it up here.
If you can pull it up, that's really
Will
important.
you,
because the overall reason why I thought this was important.
Yeah, somebody put it
Okay,
up.
thank you very much. Somebody put it on. Thank you very much. When I look at Niger, I said, for future priority research, you have to concentrate on everything. Anthracnose is very important. 120 fields out of 121 fields were infected with anthracnose. So what do? You have to have a resistant source for anthracnose. Leaf blight is another important disease for the future that they need to work on. Out of the 121 fields that we surveyed, we have 108 where we observed where the plants were infected with leaf blight. Overleaf spot is another disease that they have to look at, especially in the southern part of the state. Overleaf spot is very important, as well as roughleaf spot. For panicle disease, long spot is really essential. Resistant to long spot is very important. However, to a lesser degree, long spot was found out of the 121 fields surveyed in Niger. Long spot was found in 76 fields. However, we also encountered covered spot and head spot. Covered spot was detected in 40 fields across regions, and for head spot in 31 fields. One of the good news that we came up with when I sent the set of differentials to delimit the pathotype of anthracnose was one of the lines in our anthracnose set of sorghum differentials is PI 570-726. In Niger, this line, which is originally from Sudan, was resistant to all the foliar diseases except leaf blight. In 2 locations in Niger, this line was resistant to all the foliar diseases except leaf blight, which means this line may possess resistant genes to those foliar diseases. So you have to go ahead and integrate. Okay, just go ahead and integrate this line with some of your improve varieties or land races and study the inheritance patterns and see whether the resistant genes that you have that is in this particular line for the other foliar disease is dominant. So at least we are a little bit ahead looking at this line and looking at other diseases like anthracnose, overleaf spot, roughleaf spot. This may actually generate certain the time already that you need. Oh, not yet. Yeah, okay, thank you very much. Surveys for sorghum diseases. When we look at generally, when we talk of plant pathology, we look at the disease triangle. We have the host, we have the pathogen, and we have the environment as well. The environment may change anything. So biennial surveys or every 5 years will be essential to monitor any changes in the prevalence of the different diseases in Niger and in Senegal as well. This disease survey has to continue. You cannot stop at the end of this project. In Senegal, as I said, what I detected was that the improved varieties are resistant to anthracnose. Most of them, I can say about 95% of the improved varieties that were planted in the fields across the 7 major production areas were resistant to anthracnose, which is good news. The bad news is they were susceptible to leaf blight and sooty stripe. So you have to put that resistance to those lines that you are releasing, because just putting anthracnose resistant in the improved line is not enough. Leaf blight is very important, was the most prevalent disease in Senegal. So you have to put in resistant to leaf blight and also to sooty stripe. The other thing is grain mold. Grain mold resistant. We actually don't know, based on this survey, the importance of grain mold. I know it is important based on the few improved varieties, cultivars that I've seen when we surveyed that they were susceptible to grain mold. Some of them were actually susceptible to grain mold. However, the research that we did in Puerto Rico using the Senegalese accessions that are in the USDA collection here in the United States, actually, we had a large number of those accessions from Senegal that are resistant to anthracnose and grain mold when we planted them in Puerto Rico. So grain mold resistant and grain mold survey in both countries has to be conducted during maturity and at least before harvest. That those surveys need to be done. And finally, how do we get food stability? After knowing all this, with all the diseases, the different land races, and all those other stuff, how do we go about at least making sure that what we are releasing is actually stable and can increase our yield? It's by collaboration. You have to collaborate with other people. You can, as a breeder, if you go ahead based on the agronomic traits that you like and say, okay, this is what I want in this cultivar, in these improved varieties, these are the agronomic traits that we really high yielding, 9 scholar, correct height, and all those other stuff, and you neglect the plant pathologist, you are in deep trouble. Because what you release may be susceptible to a variety of diseases. So what you think that you will gain in yield, you actually will lose by that cultivar being susceptible to different sorghum diseases. The other thing is targeting. Targeted breeding. You can do that based on the survey. If you look at the materials that the documents that we produce based on just this one survey in Niger and Senegal, there are some areas of Senegal. For instance, let's look at Senegal. In northern Senegal, you have a concentration of long small. So I'll breed. The materials I'm gonna release in northern Senegal will be also resistant to long small. In Niger, when I look at dorsal, I look at anthracnose and overleaf spot. So in Dosso, the region of Dosso in Niger, what am I going to look for when I want to look for resistant sources? I'll also look for lines that are not only resistant to anthracnose, but also to overleaf spot and probably rough leaf spot. So I can breed materials for that particular region. So it can be targeted as well. Targeted breeding can be done with these diseases. Markets for farm produce. In those rural areas, actually, you have to create some form of, I'm talking in general of food stability in these two countries. You may have to produce, you may have to at least create markets where these farmers can sell their produce at a very competitive rate. Also the infrastructure. In Senegal, it was okay, but in some places in Niger, like just heading from what I call, was it Dosso to Gaia, which is about less than 100 miles or so. It took us 6 hours. So you actually have to have good infrastructure where these farmers can really travel to markets very rapidly. Communication and education. Most of these farmers are very intelligent. If you go there, look at their fields, tell them what's wrong, what type of diseases you are finding in their fields, and show them the neighbors, which was really amazing that some of the fields were just about less than 50 yards apart. But in one farmer's field, you will have an epidemic of a particular disease, and you walk less than 50 yards away, you go to the other farmer's field, what you will find, you find that field is completely resistant to anthracnose. So you have to communicate with these farmers. You have to actually physically go out there and tell them, let me show you. This is anthracnose. You are losing yield. Your next door farmer has a different land race, but you can see that it's not susceptible to anthracnose. So he's gaining yield. You are not. But this is education and communication with them on a personal basis by interacting with them. And the nice thing about this survey, we were able to interact with a lot of farmers in both countries. We are able to show them the disease, tell them what are the control mechanisms that you can use. Because if you want to use fungicide or something, it's just not viable, it's not feasible. So you have to explain to them using the resistant variety and also using probably some cultural control strategies to help them with some of their farming. So these are the things that I think will be necessary in the future, and it should continue. They should, the Senegalese and the Nigerians have to continue surveying these fields. If they can do it on an annual basis, they can go ahead and do it biennially or every 5 years, just to monitor what's actually going on. That's the end of my presentation. I'll invite the rest of my team, if they have any input, let them go ahead and just give it to the audience.
Thank you, Dr. Prom. That's great. So what we can do is open this up for discussion and questions.
Okay.
Before we start with you can put your questions in the chat box. You can also raise your hand. I've got both screens open and I will try to moderate that.
Okay.
I've got a question to start with, but any comments from your team before we get started with the questions? Niger, Senegal, anything to add in?
No, it's okay.
Okay, great. So from my perspective as an economist, you've done part of the job that I need in order to think about funding allocation and priorities. What you haven't said anything about is the relative yield loss associated with these different diseases. Are some of these more severe than others? Because it could be a potential misallocation if we're just prioritizing breeding on a disease to make a plant look pretty if it doesn't have any yield damage associated with it. So what can you tell us about the relative yield damage associated with these diseases?
Well, normally, when we look at the disease, one of the difficulties that we are having globally, even in the United States, one of the critical things about plant pathology is actually estimating yield loss. We've stopped doing that totally the past few decades. No funding was done to look at yield loss based on a particular disease. Generally though, when you look at anthracnose, if you have a susceptible line, you will encounter yield losses of up to 60 to 70% if you plant a susceptible line. That's a tremendous amount of yield loss. And with any foliar disease, you will encounter yield losses. How we estimate that is very difficult because there is no funding to do yield loss studies. Agencies are not going to, unless and until you do molecular stuff that they don't mostly understand, they'll give you some money to do it. But if I had money, I'm not going to, these are the things that I want people to know. Okay, let's look at anthracnose. How many yield loss do we encounter on sorghum anthracnose? We know some yield losses have been done. You know, in some areas, probably you'll go all the way to 100%. If you have a severely susceptible, if you have a very susceptible line, and in a very conducive environment, you may have a yield loss of probably up to 100%. But what they've measured so far takes you between 60 to 80% yield loss on anthracnose. On other diseases, as far as you have the photosynthetic area, if you have the green leaf being compromised, you will experience yield loss because your photosynthate will be reduced. So if the leaf is infected, definitely you will have yield losses. Probably in Africa, where they do most of those type of studies, they can tell us how much yield loss they encounter for each of the diseases. For leaf blight, if you're just looking at the surface area of the leaf, infected with leaf blight tells you, oh yes, you're going to encounter yield losses because the photosynthetic area has been reduced. So the plant is not maximizing all its resources to make seed because some of this is compromised. So you'll encounter yield loss on any of these foliar diseases. For panicle diseases,
Sorry.
excuse me.
The smell, bad smell, so that's why I call it.
Oh. For panicle diseases, any type of loss, if I planted 20 seeds and have 20 plants, and 10 of them were infected with long smut, then I have 50% yield loss. You can use that panicle. It's infected. So if you want to trash it, you contaminate the other healthy seeds. And people are not going to buy it. People are not going to buy that seed. So for the panicle disease, it's very easy to estimate. I'm at the field. There are 21 plants. 10 of the plants are infected with long smut. So those 10 plants, I have to just throw them away. So I'm left with 11 plants. So you experience yield loss that way when you're dealing with the panicle disease. You are easily able to estimate the yield loss with the panicle disease. And with the foliar diseases, Dr. Dalton, is you compromise the photosynthetic area. So you're going to encounter yield loss in all of them. But based on how susceptible the line is. If I have a line that's highly susceptible to zonate leaf spot, so if I have 20 farmers planting that, all of them are going to experience yield loss. So it's very difficult based on the fact that in the United States, I cannot document, I cannot write a proposal and say, give me $1 million and I'm going to study yield loss for this particular disease. They won't do it.
Okay, great, good. Good, thank you for that. That challenge is there. I think that you did provide some, useful insight into foliar versus the panicle diseases. Clint, do you have something to add?
Well, I just wanted to say that we've had zonate high levels in a year or two, but it's very environment related, I think.
Exactly.
But breeders don't seem to worry about it, and I don't know if they have yield data or not. I mean, it's just that they say, oh, don't worry about zonate. You can see it. It's really easy. It's the target spot stuff.
Oh, okay. Yes. Okay, Dr. Magill, you are right. You reminded me of that. Okay, in 2015 in the United States, what we had was with zonate leaf spot, if you have early spring rain, frequent early spring rain, then the disease will show up in the United States. In 2015, in Texas here, we had a lot of early spring rain, and we have an epidemic of zonate, and we experienced yield loss in 2015. In 2021, we had a similar situation in our nurseries here in College Station. If you went there in the summer and look at our nurseries in College Station, no doubt, I didn't measure yield, no doubt we had yield loss. So if you have an epidemic of zonate, in the United States, we consider it a minor disease unless and until we have situations where we have a lot of rain in the spring, then we see an epidemic of zonate. And that's what happened in 2015. In 2021, here in College Station, we have an epidemic of zonate, which means we encountered yield loss, although we didn't measure the yield because the entire leaves were compromised. The lower leaves fell off.
Okay, great, good. Other questions for the team? Anything in the chat box? I don't see anything in the chat box. I don't see any hands up.
Let me ask, say maybe one more thing. When would be the best time, or do you have to do it 4 times a year? If you can only detect grain mold and maturity and so forth, how do you know some of these other diseases weren't in the same situation that when you surveyed, they might not be so bad? So is there a good timing?
Generally, for foliar diseases, I actually go around flowering, at flowering time. At flowering time is really an ideal, what I call an ideal breaking point between what you look at later in the panicle and in the foliage. For some of the diseases, they come earlier in the season. So if you look at the lower leaves, because when you survey, you have to be very careful. You just can't look at the top of the plant and say, okay, there is free. You have to go from the bottom up to just show you what's out there. Initially, probably sorghum. Sorghum is a plant that can actually compromise, that can really, what I always call, can compensate with sorghum. Let's look at, for instance, if you go with downy mildew on sorghum. With downy mildew on sorghum, you have to reach a threshold. If I have a downy mildew in a field, I'm looking at over 30% of the plants infected with downy mildew before I panic. In the US, I'll just say, okay, it's downy mildew, it's early in the season. If that plant dies, then the neighboring plant will compensate. They will just take more space and have a bigger seed kernel weight. So there are some diseases actually that you are right, that if I see them like early in the season, I've went to a field in the US here and I see a lot of downy mildew on sorghum. I'll just say, okay, you don't have to worry too much because what I'm seeing throughout this field is less than 30% of the plant infected. So the sorghum, the rest of the plant will compensate. So I'm not gonna experience any yield loss. But what I'm looking at, in our case, what we are concentrating on is looking at resistant sources to diseases that I know, like long smut in Niger. If I go to the field and I see long smut, I know that, well, those plants really are gonna mess up if I harvest everything. So I'm gonna experience some yield loss at any time of before harvest, since they come in early, immediately after flowering. So there are some diseases when you can actually judge when is the best time. For grain mold, they have to do the surveys at maturity or close to harvest. The survey we conducted really is misleading for this pathosystem. That's why if you look at our publication, I was able to spell that out. So a person can just look at the table and say, okay, in Senegal, grain mold is not important. Or in Niger, oh no, grain mold is not important. Other diseases, let's concentrate on other diseases. The scientists in those respective countries have to make sure that they do those surveys at the appropriate time. They can't do it at the soft dose stage. They can't do it at the early hard dose stage. They have to wait later in the growing season to do the surveys.
Good, great. I see a hand up with Fana Maina. Fana, please unmute and you can ask your question.
Yes, sorry. Thank you. So the question was asked by Dr. Magill also on grain mold and other foliar diseases. So when will be the critical time for survey so that the phytopathologist will not miss, for instance, some foliar diseases? So I think you answered that. So how many times will you do the survey within the growing period? And if you are looking for a specific foliar disease, so you have to do it at a specific time so that you'll not miss that. Thanks.
Yeah, I think when I look at for zone 8, as I said, some of these diseases do come in early during the growing season and they spread upwards. So if you go look at your seedlings at a certain, for anthracnose, you can't do that. Because if you look at sorghum, the seedling stage, even if you look at in most cases, if you look at the most susceptible line to sorghum anthracnose, in the seedling stage, because of the high levels of phytoalexins produced by the crop, you won't see it at the seedling stage. You have to wait. You actually have to wait. Normally here, I wait until before I inoculate my plant, especially when I'm screening for anthracnose, I inoculate them at the 8-leaf stage. I don't want to go anything lower than that. Sometimes you can see anthracnose, in very few occasions at the seedling stage. But I wait 30 days after planting when the plant is at the, let me see, 8 to 9 leaf stage before I do my inoculations. Then I wait another 30 days. I evaluate the crop. Then I'll do it on a weekly basis for another 4 weeks. So you see, I'm starting planting all the way to the 8-leaf stage for anthracnose. Then I inoculate them at the 8-leaf stage. Then I wait again for a month before I start evaluating for anthracnose. There are papers that will tell you a snapshot, just one, at flowering or after flowering is enough for anthracnose. But for me, it's not. I usually want to make sure that I do multiple assessments just to be sure that what I'm reading is accurate. If you look at, as I said, if you look at some individuals that have worked with anthracnose, they say just at the let's say soft dose stage, just take one reading to show you their susceptibility. I'm a little bit concerned that as the sorghum plant is getting older, you have senescence kicking in, you know, their defense response is compromised. That's the reason why I want to do continual reading of several times before I say, okay, I'm satisfied with what I'm doing out here, because you can be in trouble. Let me give you a story that we had here in College Station at the University of Texas A&M. We wanted to map the CG1 gene for anthracnose. And for 4 years, they tried. I was not involved. They tried for 4 years to map the CG1 gene for anthracnose. They were unable to do it. So when I started working here, I was asked that the two teams asked me, well, we are having difficulty mapping the resistant gene for anthracnose. So they gave me the populations, the different populations, and said, okay, Luis, can you kindly help us out? So I said, yes, I'll go ahead and I'll do it. So I did the screening just for 1 year and they were able to map it. So you wanna really avoid, if you're gonna survey for a disease, the escapes and the timing. So you always wanna do it multiple times just to make sure that what you are doing is accurate, is correct. You are not missing any other thing. And another thing that you look for anthracnose too, when you survey for anthracnose, they call it the red disease. In our system, the system that I modified here with my collaborators, Dr. Magill and etc., we have a rating scale for anthracnose, which is very simple. We have a scale of 1 to 5 for anthracnose. And what we are looking for, the most important thing that we are looking for to determine whether it's resistant or susceptible is those esavoli that I showed you at the beginning, those pictures. Did you see those black dots on the leaves? That's what you have to look for. If you don't find the black leaves, you can rate for anthracnose if you are doing some epidemiological study. You say, okay, this is anthracnose infected leaves, it's red leaf. I don't see any esavoli. It's still susceptible to anthracnose, so I'm gonna go ahead and read it. You're doing epidemiological study, it's okay to go ahead that way. But in my system, if you don't see the esavoli, you say that it is resistant because it is the esavoli that erupt and splash the disease to other plants. So if you have the esavoli, the plant is susceptible. If they are absent, the plant is resistant. So you have both, you can have both a quantitative rating and a qualitative reading. We tend to go on the side of qualitative reading. Yes or no? Does it have esavoli? No, it's resistant. It has esavoli, it's susceptible. That's it for anthracnose. So always try to do multiple time assessing diseases.
Good. Thank you very much for that extensive response there. Any other questions for Dr. Prom, Dr. Magill, the team in Senegal, the team in Niger? I don't see any hands up. I don't see any questions in the box. Okay, I see one now with Mitch. Would you go ahead and ask the question, please?
So is the report going to be generally available to the through the ME, or?
A publication has been written. Is it the report different from what you published already, Dr. Prom?
No, it's not. It's not. It's not at all. It's not at all different. That's why we've based on the survey that we did, we actually made sure during the discussion to at least tell people what we are seeing and what we've observed based on this one survey of 2019. You have to do multiple surveys. You have to continually monitor what is going on in those environments. The agroecological situation may change because of climate change. So you will see different type of diseases, not with the intensity of certain diseases will come up and others will still go down, will be reduced. But you have to continually do surveys. That's what I do here in the US with grain mold in Texas. Because of the pandemic, I was unable to do them for 2 years. I still monitor what I'm seeing in infected grains here in Texas. So the report is published. So we've published, I think, 3. The survey in Senegal is published in Plant Pathology Journal. The survey in Niger is as well published in Plant Pathology Journal. And we have the other one where I sent the seeds for the sorghum differentials just to determine what races of anthracnose I'm dealing with in 2 regions in Niger. That's Maradi and Telaberi. So those that information is also published in Journal of Crop and Journal of Agriculture and Crops. So yes,
Great.
the information is in the publications.
And Jenny, you've done a great job of getting that information turned around, processed, and published very rapidly.
Yes.
Good. Other questions? Okay, Dr. Sarr.
Okay, thank you. So I just want to give a brief comment about the results we have on the survey in Senegal and the disease we focus in the project. Because I know in the project we are focused on anthracnose, grain mold, and long smut. And after the survey, we realized that in Senegal, leaf blight were more important than the other diseases. So I asked this question in the first meeting in 2020 2020 in our annual meeting. So now I don't know if we should still focus on anthracnose or if we need to move on and work on leaf blight also, because it is one of the most important diseases we have in Senegal. And our improved varieties are very resistant to anthracnose. So there is no need to keep on working on anthracnose and leaf blight like this. This is my inquiry. I don't know how we can do in Senegal. If we could still focus on anthracnose, or if we could work also on leaf blight, because if we still have samples for leaf blight here in the lab.
That's a great question. And I think that this, as the spill management has always expressed, we're really deeply rooted in adaptive management of your projects as you learn more, as things become clearer. So in terms of working on it, I guess that's a kind of a really big question. And I guess the question should be presented to the team in Senegal, the breeding team, the genomics team in Senegal, to vet that question. And so certainly, I would suggest that you bring this up, you discuss it, and you try to determine the overall economic importance in terms of yield damage and loss of other illnesses relative to what we set out to work on. So a great question, and I think that it's an open, I don't have a clear answer for you, but I think it's certainly a question which needs to be asked.
Okay, so just wanted to add something also about the yield losses you asked later. So for the yield loss, sometimes when you have a 100% incidence, it is significant to say it is a very, very, very high yield loss, because many of the plants, when they have 100% incidence, they don't give anything for harvest. Usually you don't have anything for harvest. So it is just enough to say how high our survey give us information about yield losses.
Okay, true.
Great, good point. Other questions for the team before we wrap up? We're at about 9:15 here. We've been going for an hour and 15 minutes. I see one hand up. The computer name is HP, so please identify yourself and go ahead with your question.
Oh, thank you very much. I'm Haogui Adamou from Niger.
Okay.
And I want to thank Mr. Prom and Magill, who presented the result of this work we have done together. I want to say that this work have been done in Niger in 2019, and I think that it is the first time this kind of survey have been done in Niger. So before that, we have done some episodic prospection in the country with Dr. Kolo, and we didn't have information for all the country, all the important area of sorghum production, so that this survey cover of the large area of sorghum producing area, and we are very, very this gives us good information to tackle the problem of sorghum diseases, because we have foliar diseases and head diseases of the sorghum. So I think that we'll continue to do the prospection. We have done one prospection in 2020 and 2021. We just come from the fields, and in 2 or 3 days we will go to sample for grain mold, because we have in the harvesting period. So we will continue the work like he said. Okay, thank you very much.
Thank you very much, Dr. Haogui. I don't see any more questions in the chat box, no more hands up. I want to end with one last kind of plea to the team. As you've brought up Dr. Prom and Dr. Haogui, I think you're in a great position now with what you've just described. We need correlates of the disease incidence. So I hope that in those fields you were able to at least elicit farmer identification of the varieties so we can begin to narrow search for potential resistance of materials, or susceptibility, I guess, is easier. And also those weather correlates. So having that weather information would then, you know, as we have now 3 different views of it in Niger, would allow us to better understand potential progression of different illnesses as it correlates with weather events, as those weather events correlate with climate change. So I encourage you to dig deeper and begin to catalog that information as well. So as we move forward and we look retrospectively at evolution, maybe we can have some statement on what's driving prevalence or the decline of the incidence of a disease. With that, let me thank everybody for joining us today. This is great. You've given the plant breeders a lot to think about and some great questions to ask during your annual meetings, team meetings. And I look forward to following this as we move forward. Thanks very much, Dr. Prom, Dr. Magill, the team in Niger, the team in Senegal, and thanks everybody for participating this morning, this afternoon.
All right, thank you.
Have a great day, and this will be cataloged. This will be cataloged so you can pick it up later.
Hello, Penda.
Bye-bye, everyone.
Bye.
Thanks so much.
Thank you.
Thanks.
Bye-bye.
Bye.
See you next time.